Thank you very much for the introduction. Good morning to everybody who's joining us from North America, and good day to all the other visitors that we have today. My name is Philip Bastians, and I'm going to present my talk today, expanding your scanning electron microscopy to 3 dimensions and beyond. In that talk, I hope that you would eventually walk away with understanding of the challenge to map neuronal networks. So as Gleb just mentioned, my background sits in the neuroscience or connectomics world, and this is where I'm coming from, and actually explored and got to know electron microscopy, and more specifically the 3-dimensional aspects and imaging of using electron microscopy. I want you also to walk away with some ideas of qualitative light and electron microscopy workflows, and I have presented you, or I will present one very specific workflow that I think is making the point quite clear how that can be done. Lastly, and that is a little bit of a link back to one of the previous talks in the webinar series. I want you to walk away with an insight of how we can correlate X-ray and electron microscopy data. And I hope that I can spark a little bit of inspiration to find out more. And this QR code that you see here on the slide will allow you to get to a landing page where you have access to the other webinars, and this one most likely included at some point as well. So the agenda for today is pretty much 3 parts. Volume EM in the light of, again, neuroscience and connectomics, so where I'm coming from, and I try to highlight a challenge in that field and how electron microscopy can solve that. Then next, CLEM, short for correlative light and electron microscopy, and where we combine the strength of light and electron microscopy. And lastly, with that, I will finish the talk. I will try to highlight qualitative X-ray microscopy and how we can use this X-ray data to get a targeted way to image our large volumes in electron microscopy. So starting off with a topic that I find very interesting, of course, coming from the neuroscience field, and the question why, at least in my opinion, the brain is actually a very special organ. So yeah, this is an example of a human cortex or a human brain, and eventually I want to compare it quickly to a liver and illuminate a couple of aspects that makes the brain quite special. So what might that be? Well, if we're looking at the cellular structure and maybe even look at the sheer number of cells in each of the organs, we can see that both organs are sit more or less in a somehow near ballpark number of a couple of billion cells in the liver or in the brain. Maybe there's a factor of 10 difference, and that could be already one indication how these organs actually differ. What we might add here beyond that is not only the sheer number of cells that make up each of these organs, but also the cell types that are maybe a handful in the liver. But besides the 2 big classes of cells, the neurons and glia cells in the brain, if we subdivide them into the specific classes of glia cells as well as nerve cells, you will find that there's an incredible diversity of cells. But besides the sheer number of cells and the difference in cell types, number of cell types in those 2 organs, I think what really makes the brain very, very special is the anatomy and eventually the breadth of communication, or the width of communication that each of those nerve cells can actually undergo with its neighbors. So if we compare that and do really a side-by-side comparison with how many other neighbors a nerve cell can communicate in comparison to a typical liver cell, then we will see that there's a factor of almost 3 orders of magnitude that a nerve cell can actually communicate with other cells, and that is part due to their anatomy or the morphology of those cells. And this is the next topic that I want to dive in. So if we look at the spatial scale of a neuron, we might find that one of those typical neuronal cells, which there's a pyramidal cell in the cortex, right, and the pyramid more or less due to the shape of the base and the top, so it forms a little bit something like a pyramid. Even the cell body is a little bit shaped that way. If you look a little bit closer, it's actually not that much the case. But those cells are roughly on the scale of a couple of hundred micrometers in their extent. So you can imagine that those are spaghettis coming out of the cell body. here indicated as the dendritic tree in the base and the top. And the size of those cells is already quite magnificent. What is truncated here is actually another part of the cell, which makes up the axon. And that is actually a substantially bigger challenge, because those axons are more or less the connections, the telecommunication of this purple neuron here with some other neurons. And if you follow that axon either in the brain or maybe even outside of the brain, let's focus it on the brain, maybe in a human brain, this can span up to a millimeter, sometimes even centimeters. And at the end here, a very idealized visualization here, the cell forms a connection. It's called a synapse with another cell, in that case, a more regionally cell, also a nerve cell in blue indicated here. And both of them eventually target here this gray cell. They form axons, again, a more regional cell, something that is not spanning its axons that far and further away, both forming connections here on those boutons, those thickenings with, in that case, what you're seeing here are spine heads. And those are the connections that we would like to understand. So in the field of neuroscience, there's this a subfield that's called connectomics. And in that field, the researchers that are investigating that in that area, they want to map the connections of the brain, meaning which cells are connected or getting connections from that purple cell, from that blue cell, and ideally also all the other cells that make up the synapses formed on that gray cell. So if we want to analyze that and actually have a visual representation and a way to observe and investigate that, we need to have a microscopy technology that allows us to resolve in the scale of a couple of dozens of nanometers, probably even below that, at the same time covering areas and fields of view at that resolution of millimeters, and the human brain even centimeters. That is quite a challenge. If we are thinking about this overview, millimeters, centimeters, even down to the micrometers, we can get away with light microscopy. But the challenge is that we have to detect those very fine structures that make up eventually those connections, right? So we need to have a resolution capabilities of down to sub 50 nanometers. Probably even that there are aspects of it that we need to resolve even smaller than that. So a couple of dozens of nanometers is what we need and look for. So you might think about, I would say not recent anymore, but over the last 1 to 2 decades, super resolution light microscopy comes to mind, right? And they are really pushing into that field of these around about 50 nanometers. That is great and could get you there, but the challenge is again the density of those synapses that are formed, right? So here within a very small space, we already have 2 synapses and 3 cells that we would also have to label for light microscopy. And then that's eventually the challenge. I will present you later a couple more videos and images that really illustrates how dense actually the brain is. So super resolution microscopy, even though allowing you to resolve maybe in the scale of a couple of dozens of nanometers would get you the resolution, but the challenge is you would not see everything that you need to see. So when it comes to the resolution, the real hallmark and gold standard would be electromicroscopy. So here, this is a cross-section of one of those synapses where we see an axon bouton, right, these thickenings with neurotransmitter vesicles, and this is most likely, I cannot see the whole structure, one of those spine dendrites that you see here in gray. And here, the synaptic cleft and the density tells me that this is the communication side of the chemical synapse. And here are some more reconstructions actually from electron microscopy data, and that highlights actually the challenging sides, right? Some of those axons are in their diameter get as small as 50 nanometers, and some of them, those dendritic spines, and more specifically the spine necks, are getting as close as or as small as 40 nanometers, and some even below that. So the microscopy method of choice here to really map neuronal networks is electron microscopy. So for many decades, people and researchers used a technique that is called zero-section transmission electron microscopy. And I quickly want to walk you through how that works. What we have to do is prepare a sample that is normally heavy metal stained. Then we're using an ultramicrotome, and that ultramicrotome cuts very, very thin sections, and they are floating on the water surface that is right next to this diamond knife. What you would do is you would collect many sections in series, and then eventually you have a series of those sections floating on the water. You then eventually take those sections and put them on a TEM grid. While on the TEM grid, you perform some kind of stainings. So you add some heavy metals to eventually the structures that we want to see. And then after that, you dry this off and you put that into a transmission electron microscope. And you would go from one section to the next one and find the location that corresponds to the other sections, and eventually put this data together into hopefully comprehensive and easy to analyze data set. But that's not always the case. And I hope I'm pointing a little bit to the extreme of what zero section TEM can look like. So this is one of the examples. And while we're running through the stack of images, you will see the challenges that arise. So first and foremost, all of what I presented so far is extremely cumbersome and requires a lot of manual labor. So it's very difficult to automate that. Next, those sections, since we are physically interacting with them quite thoroughly or dramatically, they shear and they twist and they deform, and all of this deformation you see in a way or two, how that eventually translates into images that are slightly deformed or warped. The cutting with a knife creates sometimes cutting marks that you see here and there striping through, or some of the sections break. And this is also something which results in loss of data in that zero section TEM data set. So when it comes to really larger volumes to comprehensively map neural networks, zero section TEM allows you to get there, but I would say in a very cumbersome, difficult way. And I would like to highlight actually how we can approach 3D large volume imaging with electron microscopy using scanning electron microscopy. So the way we want to get there, eventually, we want to have these images that I presented before in the very first slide of a synapse and this cross-section to identify those connections between cells. And what we use for that is a mode in the scanning electron microscope that I call backscattered electron mode, right? So we do have an electron column visualized here at the top. The electron beam comes down the column and eventually hits the sample. And this could be, for example, a section sitting on a silicon wafer. This beam eventually scans over the surface, and the detector here right next in between the column and the sample detects all those electrons that are emitted by the beam interaction. And while we're doing that and scanning over an area, we create an image like that. And just to quickly highlight what we're seeing here, I have a small cartoon that allows you to understand a little bit where the signals and how those signals are actually made up that we see in this image. So again, here we have our cross-section view of that sample, for example, the silicon wafer, and eventually there's a resin embedded thin section with some structure in there. And in some areas we have low C atoms. So if you think about the periodic table, there are some atoms that are of lower C, of lower N, and then eventually others that are of higher C. And when the incident beam of the scanning electron microscope interacts with each of those individual atoms, they generate a different number of backscattered electrons. And those are actually detected by this detector here. So wherever we find a high C atom, like a very heavy atom, maybe a heavy metal, it generates more backscattered electrons. And in the image here, that is translated into a high brightness pixel, or a pixel with a high brightness value. And all those bright spots here are where the heavy metals are sitting. Eventually, other areas which generates less signal is reflected here in dark. So the cytosol area, etc., there are not many heavy metals in the sample. Eventually generates a low signal. And that's how we make up the image. You might walk away here now and say, that looks like an odd image. And we like to keep things for legacy reasons in a way that helps us understand and visualize it better with what we experience. So what we do on the scanning electron microscopy side of things, we actually invert the image. So everything that is bright is now dark, and everything that was dark is bright. And suddenly this looks actually like a cross-section image from a TEM, very similar as such, right? So here's a cloud of neurotransmitter vesicles. We see mitochondria with the inner membranes, and down here that might be the beginning of a synapse. So if it would have a possibility to move in the third dimension, we might see that there's the chemical synapse eventually emerging. So the big challenge for scanning electron microscopy is that the electron beam, as indicated here in this cartoon, is interacting with the surface of the sample. So what you cannot do, what you might be experienced from or know from laser scanning microscope using light microscopy, is that you can change the focal plane, and then you look inside the sample. This is not possible with scanning electron microscopy, at least not in an easy way. So instead, we're using techniques that allow us to open up the third dimension. And those are indicated in this slide here. So what we see here on the left is a heavy metal stained brain. That is actually a mouse brain. You might see the olfactory bulb right here in the front. And these are thumbs to give you a little bit of an idea of a scale of how big that sample is. It is very black due to the heavy metal staining. And eventually, this is what we need to create images and actually volumes of data, as you see here on the right. You see here blood vessels. These are actually cell bodies of neurons. And you can sometimes see, a long stripe that goes from bottom to top. These are actually apical dendrites, right, from the pyramidal cells that I showed in the beginning. So how do we open up this third dimension to actually acquire these three-dimensional data sets? So far, what we have seen from the scanning electron microscope data, it's everything in 2D. We can, very similar to zero section TEM, we can use a technique that is called array tomography in the scanning electron microscopy world, where we again collecting those sections and deposit them on a coherent substrate. And I will, in the following slides, I will go into much more detail how that actually comes about. And then we image one section after the other and create from each of those images, we stack them on top of each other, a three-dimensional data set, as you can see that here on the right. An alternative here can be in the microscope chamber mounted ultramicrotome. This method is called zero block phase scanning electron microscopy, where we have a diamond knife here, a block of tissue, for example, from that mouse brain here, that cuts a very thin section off the top, and then eventually our scanning electron microscope here, indicated as the cone on the top, would scan over the surface to create an image. Then a cut would appear, and eventually we would, over time, put together a data set, just like the one here on the right. Lastly, we can use focused ion beam scanning electron microscopy, and you can just imagine when it comes to this technology that you're replacing the diamond knife with a focused ion beam to mill into this third dimension. Now, again, in the following slides, I will go into much more detail how that comes about and how that works. So let's focus first on the zero block phase technique. I brought you a video where we are flying into the big chamber. It's also something very, very different compared to a TM. We have actually a huge space in that vacuum inside that chamber, where we have an ultramicrotome mounted. Again, here's the illustration or indication of the diamond knife. This would be the bottom of the electron column. And right here on the top of that aluminum pin, this would be our sample that we want to image. So the knife just moved out of the way to reveal the surface to eventually the electron beam. We scan over that surface, and then each of those backscattered electrons that we see here are detected by a detector up there and creates an image. Next, we stop the imaging, and eventually the diamond knife moves back. We move the sample up by a certain step size, and then a small section is cut off. This is a bit idealized, so the section is pretty much gone, so it's literally wasted, it falls apart, but the new freshly exposed surface is what we are scanning off. And this we can do hundreds, if not thousands, or even 10,000s of times, very, very automated, to eventually put together each of those images into a three-dimensional data set. So the data set can look like this example here, and here we are using or a proxy to actually investigate the third dimension or the three-dimensionality of that data set. So we are actually always seeing only a single 2D section, but we replace the third dimension with time. So we are running through the data set back and forth. So we can see actually the 3D structure emerging and disappearing if it's, for example, something spherical like those mitochondria that we're seeing here. And from time to time, if you look very closely, and you could slow it down, of course, but here, this is, for example, a synapse, and we can see myelinated axon here with this big, thick and very dark myelin sheet around that, to eventually analyze now by the structure that we're seeing, which of those cells are making connection with each other. Not only that, so this is only one view of that three-dimensional data set. Those two stripes that we see on the side and on the bottom are actually giving you a completely different orthogonal view of that data set. So in that case, at the bottom and on the top, we are moving in the perpendicular to each other, to each of those planes that we're seeing through the data set. So you can analyze and view that data set in all arbitrary axes if you want. And that's what's really making this data extremely rich for analysis of three-dimensional mapping of eventually the cells and those neurons that form connections with each other. So next, I want to cover the focused ion beam technique. And here we are using a focused ion beam that is mounted on the chamber here on the side, and eventually the electron column would be the top part here. So this is indicated in this sketch here with the green cone, and eventually a cell that sits in the chamber and a target, right? This ellipsoid that we're seeing here that we want to image. With the focused ion beam SEM, again, we have the second column sitting here on the side that can form a focused ion beam. And the biggest difference between the scanning electron microscope and the focused ion beam, or the two columns and the beams that we generate onto the same spot on the sample, is that the scanning electron microscope or the electron beam we use for imaging. The focused ion beam we use literally for ablating material. So wherever the focused ion beam hits the surface, it removes material at that location. And this is something that can be very, very precise. And I will show you a technique how we control actually the focused ion beam in our systems. So what we do first is we are trenching into the surface and creating a new perpendicular block face, right? So originally the block face was the long edge here, but now with the focused ion beam, we are generating actually a trench in, and this trench, this side wall here is actually what we are imaging. So the scanning electron microscope comes and scans over that block face here on the side and creates an image like that with an onset of a cross-section going through this ellipsoid volume of interest. We can then use the focused ion beam again in a consecutive step to trench in a little bit further, maybe a couple of nanometers, maybe a couple of tens of nanometers. And then we use the scanning electron microscope again to image that block face, that newly generated block face, and we save that image on our computer. And then we can do hundreds, if not thousands of times. And then the last step eventually is again to take each of those 2D images, stack them on top of each other to recreate digitally this ellipsoid volume of interest. So I mentioned that the focused ion beam, you can replace that, or this is pretty much our replacement for the diamond knife. It's a very, very precise ablation tool or mechanism that we can use for this kind of acquisition. So if that is our sample, again, we are creating a new block face. The focused ion beam trenching goes into the red arrows direction, and the scanning electron microscope scans off this block face. And for this technique, it's very important to keep track of where's the focused ion beam actually ablating away material. And for that, there's a very neat technique that we call Atlas 3D, where we are depositing a platinum pad right on that block face, right where the red arrow is sitting on. And that appears in our scanning electron microscope in the cross-section that we're seeing here, very, very bright. Now, on top of that, we can use the focused ion beam to trench in these trenches, these beer claws, that you see in the middle here in the cross-section, just as chevrons spiking down. And the two on the side are milled at an angle. Eventually, while we are scanning and ablating away this material here, we can see that those three trenches in the middle, they stay at the same location, and the outer ones, they're actually moving inside. And that's very easy to explain. While we are moving in here, right, again, this in that direction would be up in this image. Those trenches here on the side, they will come closer together. And we can measure how far they progressed and closed in. And by also knowing the angle between those two trenches, we can very easily calculate how much ablation progress did we actually do in the last ablation. And why is that important? Well, our focused ion beam is not always that precise. Meaning sometimes it's milling away material faster, sometimes it's doing that a little bit slower. So this here is a feedback loop that allows us exactly to measure how much the focused ion beam actually mills in. And down here, I hope you appreciate that this is again inverted neuronal tissue that we image, like with all those bright spots. The round ones are mitochondria, the dark ones are probably big dendrites, etc. So this is something that we have published. And it allows us this kind of observation tool to consistently mill in the sub 5 nanometer thickness. So what you see here in that graph is the slice thickness versus the slice number. In the beginning, I always call this a little bit the equilibrium phase. So after around about maybe 2-3 dozens of sections, we are getting eventually to the set slicing thickness, and that we can do very consistently and monitor and correct for over many of hundreds, if not thousands of sections. So this is something that can be done very automatically. So now I'm trying to bundle everything together in this very nice animation from a couple of friends at the NIH. So again, we are depositing pad of platinum on top of our block face, right? We are milling in those trenches here with a focused ion beam. We put some carbon on top, and this is really important for the contrast that we're eventually seeing. And only then we start to trench into our sample. This can be done very fast, or we can do it in a polishing step with a focused ion beam to have a very smooth surface here of our cross section, and that's in that case a single cell. We use the scanning electron microscope to scan over this block face, and then we do, just as I mentioned before, the ablation and imaging workflow, and put in the last step, when the experiment and the ablation is done and all the images are on your hard drive, we put those images together into a 3-dimensional data set. What we cannot do on top is using those chevrons that are sitting at the top as registration marks that allows us to do this registration eventually and pulling out 3-dimensional data set fairly automated. So as the last method, I want to go into the array tomography method. Again, we are starting off with this heavy metal stained sample here, this mouse brain sample. And we use a tool that's depicted here on the right. There are a couple of vendors. I don't want to highlight anyone, in that case, that's from RMC, an ultramicro that allows you to cut very, very thinly sections of your heavy metal stained sample embedded in resin. The heart of this ultramicro is actually right there. And here, this is an inset of that. And what we can see here on top of that is a diamond knife with an attached boat, like a small water cup attached to it. And the sample would be sitting at that long lever here. Eventually it's hidden here, but I have a video in a second that really allows you to appreciate how that actually comes about. So here, this is actually the similar tool. In that case, we have attached a slightly different scaffold that allows you to put a silicon chip inside that water bath that I mentioned right there. Right, we are positioning that under the water surface, and right here, this is the diamond knife eventually, and that is the sample, right? Again, black, you can see the comparison here, black here, black here. Put a little bit of super glue on top of that so that those sections that are cut at each iteration are actually sticking together in those long ribbons that you just saw. When this is done and the water surface is covered with all those long ribbons consisting of many, many sections, we pull out that silicon chip and eventually place that on the surface, let it dry off, and then it's pretty much ready for imaging on the electron microscope. This allows you to do a couple dozens, if not a couple hundreds of sections, but there are methods out there, and that's why I call this array tomography method a very versatile tool that allows you to collect actually thousands, if not 10,000s of sections. And this is something that I just want to briefly highlight. So instead of using that silicon chip embedded or underneath the water surface, we're using a tape system that continuously collects those sections one after the other. So there's like a conveyor belt that whenever the next section is ready, picks it up and puts it onto this tape, and this then eventually goes onto a tape storage wheel, and from there we mount it on silicon wafers, right? So we unroll the tape, we put it and glue it onto a very flat surface, and a 10-inch silicon wafer is an ideal substrate for this, and mount it on top of that, and then you can probably imagine that we can have many of those silicon wafers, and then we can store them literally in libraries of already cut brain samples that we then image on the electron microscope. So all of that is just pretty much the thin section preparation that eventually allows us to have a sample ready for electron microscopy. So how do we do the next step of imaging? Because that can be done easily or a little bit more cumbersome if you do it manually. So let me show you an interface and workflow that is based in Atlas, and more specifically Atlas array tomography, where we can work off a photograph, literally with a photo camera, of that silicon wafer with all those sections here indicated in green in an interface that allows us to navigate on with the electron microscope using this photograph as a navigation map. So in the first step, we would need to tell the scanning electron microscope and eventually the stage inside the scanning electron microscope, where are all those sections? And the way we do that is, again, slightly manually, but you will see in a second that this can be done fairly quickly. We draw the very first section, a reference frame around one of those sections. Eventually, there's a tool that allows you to use that first section as a template and then snap it on the next one. Those sections are barely changing over the course of a couple of sections. So they look very, very similar, at least on those tapes here. And then you can move and click each of those, and this tool, this template from the very first section, eventually snaps onto all of them. And with a minute, make it 2, you have pretty much told the electron microscope where are all the sections. In the next step, we would take a first image. Normally this is done fairly low resolution. So down here, you can see the icon or the letters protocol, where you can define an imaging protocol. That might be high resolution already or low resolution to just grasp an overview. In that case, this is a sample that is collected in that ribbon format, not on the tape. I just used it as an example that this interface here allows you to do both. The tape collection system or these ribbons sitting on the silicon wafer. Here we imaged all those sections already in an overview manner, and then eventually what we eventually want to do is having high resolution, like on the scale of nanometers in that area. What we can do to really save time is to highlight from that overview images those areas that we really want to image in higher resolution. And this is automatically done for what the site that you're generating for one section is automatically propagated to all these other sections, so you don't have to do that manually. This is something that it really allows you here to automate the workflow as much as possible. In the last step, you eventually have then your high resolution data, and then you can observe it in that interface, whether the data is usable the way they are, or it appears, or if you have to re-image maybe at a higher resolution, or something went wrong or interrupted your workflow, you can re-image the same section to have it in a pristine data quality. The tool here does also allow you to correlate the single sections with each other, and eventually allow you to have a data set ready that is aligned and cubed for some other software that you might be using to visualize it in 3D. In that case here, we're using software from ORS that allows you to crop the data and use the cross-section and highlight some of the aspects here in 3D. So that brings me actually to the next step. Why do we have to use or limit actually the area that we want to image? Well, it's a fairly straightforward problem. Our electron microscopes, since they are scanning electron microscope, they take some time to scan over a certain area. And let me illustrate that with one of the holy grails that back then when I was still in academia, we went out to actually gather, and that is a 1 millimeter cubed data set from the brain. So if we cut this down with the array tomography workflow and collect, for example, 50 nanometer sections, and eventually calculate up and sum up the area that we have to scan off, we end up with an area of around about 20,000 square millimeters. So why is this important? Well, we can use a frequency, how fast state-of-the-art scanning electron microscope needs to scan over this area, right? So for an area of roughly 1 square micrometer, a state-of-the-art SEM needs 2.5 hours if we apply a pixel size of 4 nanometers, right? And then with this number of 20,000 times 2.5 hours, we can do the calculation very easily, 2,083 days. So a cube millimeter would end up a scanning electron microscope to have to scan 24/7 for 6 years. That's not very enticing. That was actually quite a big challenge. You can venture out, and some PhDs will probably do that, but it's not very reasonable to have a system running for 24/7 for 6 years. So that is something which was a big problem. Around about now almost 10 years ago, a very probably known researcher at Harvard, Jeff Lichtman, partnered up with SAIS to think about the technique, how to overcome that and how to reduce the time that takes us to scan actually a cubic millimeter in more reasonable timeframes. And we came up with this tool that you see in the background, right? Jeff Lichtman, he's, I would say, a good-sized man, but this tool is quite a massive box, and eventually allows you to do that job substantially more easily, right? And we can reduce the acquisition time now to 3 to 4 months. So in less than half a year, we can acquire now a cubic millimeter, which is still a daring task, but substantially more feasible in comparison to 6 years. So how do we do that? Well, we're using multiple beams that scan over the area that we want to image, right? So, and this is just an overview and sketch of how the technology pretty much works, like very high level overview, and feel free to read it up with that publication that I put here on the bottom right. So again, we have a multi-beam source that eventually runs through a beam splitter, which focuses the beam, and more specifically the beamlets, onto the sample. And what is very important is that to understand that each of those beamlets pretty much interact with the sample, just like I described it in the first place. And there are electrons coming out of this interaction. And the important part is to keep those signals from each of those beamlets separate onto eventually the detector. So those electrons are now moving up again through the column, through the beam splitter, onto a projector, and eventually a detector. And that allows you pretty much to increase your throughput, multiplied by pretty much the number of electron beams that you have available for parallel scanning. And all of that is, I think, best understood with that small animation here, where we're comparing a single beam machine with a multi-beam scanning electron microscope. So again, here's our beam splitter that allows us to separate that beam into many beams. In that case here, it's 8, I think, if I'm not mistaken. No, it's actually 7. And those are scanning over the surface, and then each of those interactions at the surface will create a signal stream, right, in green, that gets projected onto our detector, right? And that right away allows you to understand that each of those beamlets scanning over the surface creates an image in parallel, right? And we can increase the number of beamlets now by 61 beams, or there's a second machine that we have 91 beams, and that right away gives you the increase of throughput that you can actually easily calculate. But it's not only that, we applied some more tricks to not only scan in parallel, but also way faster. So that allows us really to bring down this acquisition time quite dramatically, right? And if we assume we have the same scanning frequency, then you would just get 91 or 61 times as much area imaged at the same time as one standard scanning electron microscope covers one of those tiles. Each of those hexagonal final images or single images we need to put together eventually into a 2D image that is quite fairly big, and then eventually we move on to third dimension, and then we'll highlight that in a second. So again, with an image is now 100 by 100 micrometer at a spacing of around about 4 nanometers, and we can scan extremely fast. So this is 100 nanoseconds dwell times, corresponds to around about 10 megahertz. That really allows you to scan very fast, which is in that case not that possible with a single beam machine. So it's not only the parallelization, but also the faster scanning that allows you to get there. Large area, and this is what I really could consider large. So we're talking here about around about 2 millimeters by 1.5 millimeters cross section. We can image in pretty much 6.5 minutes. That's really fast. So this would take you very long on a single beam machine. And eventually, this whole workflow looks as such that you acquire a single image now in a hexagon style. You 2D register each of those hexagon into a 2D image. And this you do for each of those sections that you image. Then the next step is to 3D register it. So you take one of those stitched 2D planes and align it with the next one. And after you have done that, you have your 3D reconstructed data set, in the case now, this idealized 1 millimeter cube. The biggest challenge of those workflows now, at least in my eyes, is to make sense of the data. I think the bottleneck of your electron microscope generating these 3D data sets is pretty much solved, and it's quite mind-boggling how much data they can actually pull out. The biggest challenge and bottleneck at the moment is segmentation and visualization, actually make sense of the data. And that is actually where I would say the dramatic majority of the focus is sitting at this moment. Here's just an overview of how big that data can be, or at least this is an image from the cortex, and you see those pyramidal cells are in different colors, and eventually there's a data set that I will show you in a second. highlighting a very densely reconstructed portion of that data set, right? So each of those single cells here are, most of them are pyramidal cells. I saw the one that's actually not a pyramidal cell. And eventually this publication, I would say this is really a landmark publication from 2015, put the small tube, cylinder here in a densely reconstructed data set. And there was some analysis done, which was fairly interesting. So let's quickly compare these methods. Again, I would say there are 3 methods available. They differ in certain extents, and I want to quickly highlight what are the differences. And I will map all of them against the zero-section TEM technique that I illustrated in the very beginning. We are using the 4 dimensions, and again, there are probably more considerations, more aspects, how you can cross-sect and compare those methods. But we use pixel size, or the eventual resolution that we can achieve, the slice thickness, so how thin can we cut, that allows us to actually get an idea of how good is our 3-dimensional resolution, how fast are those methods, and what is the potential acquisition volume that we can cover. So when it comes to zero-section TEM, we will achieve the highest resolution possible from all those techniques. But at the same time, the slice thickness is very limited, and the acquisition volume is limited by the amount of work that you have to put in, especially the manual work. And the speed is also fairly limited, putting everything together, the imaging, and eventually having all those sections put into the electron microscope and the data then processed afterwards. If we compare that with focus ion beam SEM, you will see that the resolution drops a little bit in plane, but the slice thickness dramatically is reduced. So a typical slice thickness in a TEM sits probably at around 70 nanometers. Maybe there's some really skillful ultramicrotomists that can cut 50 nanometers or even below, but that's very, very challenging. The slice thickness on a focus ion beam SEM can be as small as 5 nanometer or even smaller than that. It really depends a little bit on the sample quality. It's also very, very fast. So everything happens inside the electron microscope. You don't have to take it out. You don't have to manually interact with the sample. The focus ion beam milling and the imaging is automated as much as possible. And we can fairly acquire definitely a larger volume than what zero-section TEM allows you to do. If you want to acquire even larger volumes, then zero block phase SEM is something that you should go for. The resolution is a little bit reduced. That has to do with how the sample is put together. So we are really scanning off the block phase here, and that is a little bit of a challenge. The slice thickness is not as good as focus ion beam SEM, but dramatically better than what you can achieve with zero-section TEM. And it's fairly fast. So both the cutting and eventually the area that you scan off is something that is substantially faster compared to a focus ion beam SEM. Last but definitely not least is array tomography, and I use an illustration here in combination with the multi-beam. So again, the slice thickness is probably much on par, if not slightly better than what you can achieve with zero-section TEM. The resolution is very, very similar to the TEM resolution. So you can go actually fairly small. And again, always keep in mind, I'm illustrating that against resin-embedded heavy metal samples. So if there are other samples that might look slightly different. The acquisition volume is, of course, massive. And when it comes to speed, this is second to none when it comes to throughput. Comparing all of them, you might be asking, we have all those techniques, but why doesn't the field converge to one? Well, there are different research requirements for the data set, and eventually that allows you to eventually answer your scientific questions. And depending on what you need, the data set volume, the resolution, you might favor one method over the other. So for example, FIBSEM tomography is often used for single cells, sometimes for very, very localized areas in the brain. But a single cell acquired with FIBSEM is often the best combination between, we want to have the highest resolution, and the volume of a single cell is so limited that focus ion beam gives us really the best quality of data in the third dimension as well as in the block phase dimension. So we want to go for larger volumes, and I would put more or less a benchmark here of less than half a millimeter, cubic millimeter square, cubic millimeter. It might be best put into the block phase, the zero block phase area that allows you to very, very straightforwardly acquire the data and eventually capture a good area of again, a couple of 100 micrometers cubed. And largely, if you want to really go beyond that, I think array tomography is the only way to go, combining that with the multi-beam technology. I quickly want to highlight 2 publications here that allows you to get a little bit of an idea of what has been done with these techniques in the neuroscience field. So first of all, here, this publication from Helene Schmidt et al. Something that I consider very, very interesting. And here we are looking at the entorhinal cortex. So that is one of the cortices that are interacting very, very closely with the hippocampus. And you have the so-called grid cells in there. And what Helene et al. Were doing is they wanted to look at local circuitries that are happening inside that brain. So there are different modalities how signals are propagating through a local a neural network. And what they did is actually they were able to identify some kind of synapse sorting, where an excitatory cell that gets excited from an external input activates first an inhibitory neuron or an interneuron that acts as an inhibitory signal down the road, and only then later the synapses are formed on other excitatory neurons. So eventually this inhibitory neuron comes a little bit delayed, but shuts down all the activity down to all the other activated neurons from that very first cell. And that is something very, very interesting. And actually Helene and I went in and analyzed the synapses, really reconstructed them by going into the electron microscope data and finding out, okay, this is a chemical synapse. And putting a connection from the purple cell to the black cell in that case, and all the other excitatory purple cells later on, and she can measure more or less the distance of those synapses, and all of that is really the richness of the data that allows you to do this kind of analysis. Another one of the publications that I really want to highlight is from Fabian Zwara, who went into the zebrafish. It's a quite famous model organism, and he went into the spinal cord and acquired the data set here with zero block phase SEM. He went in and wanted to investigate the circuitry behind those big, also again in purple, motor neurons that drive the fast swimming motion. And they went in and reconstructed the cells and also identified the interneurons here and hypothesized 4 circuitry motifs that might drive those big motor neurons here in purple again. And eventually they found out that this dorsally fast excitatory interneurons are the ones that are really driving by the circuitry and the measurement of the synapses, those motor neurons. And again, something I want to highlight, most of those publications are really based on human annotators or students that reconstruct the data. So this is really one of the biggest challenges, and slowly the field is moving on to reconstruct the data in a bit more automated way. So let me move on to quickly some correlative light and electron microscopy. I'm running a little bit out of time. And I want to highlight again this data set that I presented here from Harvard, from the Lichtman lab, and Bobby Kasturi was actually the lead author here of that publication. And now we are seeing this small cylinder, and again, this is reconstructed densely. So all the structures that are inside that small cylinder, and just to put a number on it, it's around about 12-15 micrometers in length. And all these details that you see, this is actually the richness of the brain and all the structures inside. So light microscopy would not get you there. This is really something that is you can exclusively only do with something like electron microscopy, where you see all the membranes and eventually all the processes contained in there. What they went on is not only reconstructed all the structures, but use local features in the electron microscope data to classify what kind of structure are we looking at. There's a bucket of axons, myelinated axons, excitatory axons, inhibitory axons. Here in the middle, they highlighted the glia cells, right, astrocytes and oligodendrocytes. And then on the right, they have different types of spines, so dendritic structures, and they focus the data set on those 2 central spiny dendrites, right? You can see that it's a shaft, and then all these small bubbles and stubs that are moving away from that shaft are actually the spines. Then there are other spiny dendrites, and then there are smooth dendrites, which is very peculiar to see and often a hallmark of interneurons. But they also highlighted that sometimes they were not really able to identify all the structures by the local features. And that is, of course, a challenge. So if you would go into this data set and would ask yourself the question, hey, how can we improve this? How can we avoid that we have those unidentified structures? You might think about, we could we could bring in some prior knowledge. And the way we can do that is use light microscopy. So here I brought you an image from a light microscope where we highlighted with a tag, a fluorescent tag. However, this is presented to the cell, and that is eventually imaged with a light microscope. But as you saw from the video just now, this is a very arbitrary and quite a distorted view of where this spiny dendrite in this case lives. So this is embedded very, very densely in other structures. But light microscopy, as great as it is, we can only see what we are highlighting. So that is the shortcoming, but at the same time, the strength, because what we are highlighting, we have a very, very good understanding of what it was, what we were using to tag that cell. And this is very, very exciting. But the second you overdo that a little bit, well, then you end up with an image like that, where it's very, very difficult to actually see all the structures and allocate the right dendrite to the right cell body. So there are strengths and limitations on both sides. Electron microscopy, despite us being able to cover huge volumes, is very limited still when it comes to identify all the structure. And light microscopy, you can pretty much only see what you're highlighting. So you already brought in some prior knowledge, and ideally you want to combine both of that. And that is what correlative light and electron microscopy is all about. So we want to put things into context. We want to find rare and target events. And this is really something that comes out of the light microscopy world. And by the end of the day, we want to gain new information. We want to be able to answer scientific questions that we haven't been able to answer before. Let me drive you through one of the correlative light and electron microscopy workflows. And just keep in mind, this is not the only one. There are probably as many workflows as you could imagine, and then some. But this, I think, is a workflow that's very easy to understand. We call it SEM correlative array tomography, where again, we are using a substrate that allows us to collect those ribbons that I showed you before from the array tomography workflow. So those white stripes, just imagine those are ribbons consisting of many, many sections. We can then apply some fluorescent staining onto those ribbons and those sections, and then move that into a fluorescent light microscope. Image each of those sections, put them actually together into a three-dimensional data set if we want, and choose to already at that level. And then we can go back and rinse out these antibodies that we have used and maybe add another antibody. And then we do the very same. We image again with a fluorescent microscope. So you can actually create a couple of three-dimensional light microscope data sets and use that later for the correlation. As last step, we are introducing the heavy metals. And this is really important that we do that last, because there's some really bad interaction between antibodies and also the antigens that those antibodies might bind to with the heavy metals. So it's important to keep that for last. And then we move into the electron microscope and image the same sections again with the electron microscope. The software interface here allows you to visualize both and put together the correlation. If you have ever worked with something like that, it's never as easy as I'm demonstrating it. But our workflow and this correlative array tomography workflow, we try to make it as easy as possible. So where do we know where those images, again, on the scanning electron microscope are taken, and where those images have been taken on the fluorescent light microscope? Well, we are using a trick. And those are the 3 fiducial markers that you see in the cover slip here, and that you see in the cover slip here, right? Indicated as 1, 2, and 3. And before you take any images on the fluorescent light microscope, the wizard here, or the workflow, forces you to image those fiducial markers first. And it does so on the light microscope, and it does so on the scanning electron microscope. Meaning you have a reference frame of all the consecutive images that you have taken from the fluorescent microscope, and you have taken from the scanning electron microscope. And this reference frame allows you to very straightforwardly correlate each of those images as conveniently as possible. I brought you an experiment here of one of those examples. Again, these are thin sections of a HeLa cell, and in that case, we use the DAPI stain for the nucleus, and eventually huntingtin here in red. And as argued before, we can only see what we marked. We see the nucleus and this huntingtin, but nothing much else. Again, on electron microscopy, we see all the structures that actually bind up heavy metals, and of course by the structure we can indicate this is a nucleus with a plasma membrane. There's some philopodia maybe from those HeLa cells, and all of that can be visualized. And with the SEM workflow, we can overlap and superimpose them. So each of those sections that we image on the light microscope and the electron microscope, we correlate and superimpose the data. And lastly, we can use each of those sections to correlate them on top of each other. and then create a three-dimensional qualitative light and electron microscopy dataset. So if you have a 3D light microscope dataset and a 3D electron microscope dataset and just want to correlate them de novo in that 3D state, it's very, very challenging. But this workflow here allows you to divide and conquer. You solve this problem first in the 2-dimension, and then you correlate that into the 3rd dimension. And then lastly, you can, of course, visualize that and then highlight the fluorophores and the target that you are identified on the light microscope in context of the structural information of the electron microscope. So I hope that gives you a little bit of an idea of how you can do qualitative light and electron microscopy. And I want to segue to the last topic, correlating actually X-ray microscopy with the 3D SEM techniques that I highlighted before. Just to illustrate a little bit the problem that I haven't mentioned before, but that is very apparent to everybody who has done 3D scanning electron microscopy or volume electron microscopy. Well, normally we start with a very, very big sample. Like that can be on the scale of centimeters even, or millimeters, but all of that, of those two scales, even the millimeter level, it's still very large volume for electron microscopy. Typically, we cut down a small portion of that brain in that case, maybe a millimeter or 2 millimeter volume that we want to eventually image with one of those 3D SEM techniques. And then lastly, we might end up with a data set that is cubed and the edge length around about 15 to 100 micrometer, just as one example. That looks extremely straightforward, but it's actually more cumbersome than you would think, because normally you already have an idea which which part of that brain you actually want to image. So let's turn this around. Let's start off first from the region of interest, or your volume of interest, is probably a better term, and think about how that is buried actually in your sample and your whole sample preparation. So there's a tissue block, and somewhere in the center, ideally in the center, or maybe even distorted closer to the borders, there's your volume of interest. And let's assume a tissue block of around about 2 millimeters. So when we have a 60,000 times larger volume, then eventually the final volume of interest. And to make matters even worse, all of that is embedded in resin. And resin is it's really cumbersome the moment it's cured. You can't really see any structure anymore inside your tissue. You see the overall structure of your tissue block, which is very, very dark, but you will see in a second, it's very distorted, so you can't really see inside. And what I want you to be aware is that there are a lot of those examples that end up with a sample that we see here on the screen, where we approached the sample with focused ion beam SEM and trenched into various regions, not really knowing where's our volume of interest. And I can tell you this is around about a week worth of work. And you don't want to be the operator walking away, not being able to demonstrate that you actually found your target interest, and probably even have to abandon the sample because you just don't know where you are. So let me provide you with a work that I did with Dr. Isabel Bon here from the National University of Singapore, where we were looking into a heavy metal stained, resin-embedded mouse lymphatic tissue preparation. And here, this is one of those typically embedded resin blocks. And then the structure here is the dark stuff is actually our target. And this can be, again, on the scale of millimeters in the worst case. So we wanted to apply zero block phase SEM. And what I would normally do is I need to trim down that sample to maybe a block of a millimeter cubed. And blind trimming was not really an option for me because I had only one sample and I didn't want to waste it. So I want to really make sure that I hit the right volume of interest as precisely as possible. So how could I do that? If you follow the talk from one of the previous webinars from Rosie Manzer, she probably told you everything about X-ray microscopy. And I just quickly keep that sketch here to quickly run through, but in a very high-level overview. You have an X-ray source, you have a sample in the center, and then a detector on the right side, and something that SEIS does very, very well. We can magnify that even further. So we rotate the sample, and each of those rotation steps, we take another image, and this we can combine into a three-dimensional data set. And what you need to take away here is we don't destroy the sample in that process. So that allows us to do something like that. Again, 5 millimeter sample, and then eventually we can illustrate that and view that three-dimensionally from the surface, and then we can cross-sect and the sample and eventually run through and preview what is actually inside that tissue. And again, we wanted to see lymphatic vessels. And I thought, this is great. I mean, I can see it. It's running right from the top to bottom I looked at the data, it was very clear to me that this is the one. But at the same time, also acknowledge I'm a neuroscientist, not somebody who's working in the lymphatic or blood vessel system. And the research actually came back to me and said, well, this is actually a blood vessel. That's not a lymphatic vessel. The vessel you are looking at is actually the small one down there. And by the way, there's another one up here. So I was only able to identify those small targets by having this preview with the X-ray microscope. Right. So what we did next is we cut down the sample and pulled out that small block, mounted it on a metal pin. So this is actually the sample, right? And re-scanned it, right? So the first sample we scanned was this one, the second one was this one. And this is what we got. So again, the block, we're running through top to bottom, and right there it is, our lymphatic vessel. So this is what we wanted to target. And we went in with the Scanning Electron Microscope and took an overview image. It's a very, very large one, 600 by 600 micrometer. So we can also take large overview images. And you don't have to believe me, you can convince yourself whether this is actually the same region from the X-ray microscope on the right and the Scanning Electron Microscope side on the left. And I try to highlight a couple of structures that you might recognize in both images because it looks a little bit different. But it's bright on the X-ray side, it's actually very dark and black on the Scanning Electron Microscope side and vice versa. But if you look at that vessel right here, right, you can try to point out some of the structures that you might recognize. And then eventually this ripped through one of those lipid cells, right? You see that here as well. And then there are some more markers here, and then there's another tier here and here, right? So I have with the X-ray data pretty much a preview of where I will be cutting into my sample with the zero block phase SEM technique. And that allows me really to very precisely tailor where do I need to put my field of view that needs to be scanned off, how many sections do I have to cut until my structure emerges, and really keep an overview and already a foresight of where I need to acquire my volume. And not only that, I can also later take this data set and correlate it with the X-ray data. That captures the overview of the whole sample that has been cut away in the imaging process on the Scanning Electron Microscope, and I can maintain the overview from the X-ray data, and that allows you to keep your navigation and orientation of where was your Scanning Electron Microscope data actually acquired in the context of the whole sample that has been acquired with X-ray microscopy. And of course, yeah, this is, sorry for that, this is three-dimensional data. So we're running through here the data set and see how the vessel eventually moves here to one of the borders of the images. And this is, of course, very low resolution. But at the end of the day, we want to have high resolution data set. So we image here one of the borders of that lymphatic vessel. And what the researcher here was very interested in is to see the collagen fibers that are running all around that lymphatic vessel. Right? There's an endothelium, there's some smooth muscle cells, and then eventually you see all those fibers running through the data set. And this is what they were really interested in understanding how some of the diseases actually affect this lymphatic collagen fiber network, and causes to have some of those lymphatic vessels to be more weak than in a healthy state. So with that, I'm really sorry that it took a little bit more time than I was expecting. I have a quick summary, and again, this is what I would really like you to walk away with, to have a little bit of an understanding of the challenges that the neuroscience and more specifically the connectomics folks are facing to eventually map neuronal networks locally or even in the whole brain. I hope you get an idea of one qualitative light and electron microscopy workflow and the overall concept, why this is attracting and interesting for researchers to apply that to their electron microscopy data. And lastly, I went into a very specific challenge that I faced also during my PhD and still on now at SAIS, and that is finding the right volume of interest in my data set, in my tissue that I prepared for electron microscopy, and am able to tackle that with correlating X-ray microscopy with eventually the electron microscope data that I'm acquiring. And with that, I hope you enjoyed the talk. And Gleb, I think I give the word back to you and see if there are some questions.